All life reproduces. Of all the hallmarks of life, this one is likely the most obvious to most of us. Reproduction is done either sexually or asexually. Though certain images may come to mind when one hears the term sexual reproduction, sex is not necessarily involved for sexual reproduction. What is involved, however, in sexual reproduction is the mixing of one's parent's DNA with the other parent's DNA. In sexual organisms, the DNA is transferred in gametes, sometimes called sex cells. The sperm and the egg are gametes in animals. Pollen and egg are the gametes of many plants. In most animals, of course, sperm must fertilize an egg for reproduction to be successful, but having sex is not necessarily involved. For example, some male salamanders deposit a ball of sperm called a spermatophore under the ground, and the female will pick up the spermatophore with her cloaca, the beginning of her reproductive tract. So you see, sex does not necessarily occur for sexual reproduction, but a mixing of male and female DNA must occur. Asexual reproduction means without sex. Specifically, this method of reproduction does not involve gametes. While most organisms reproduce sexually, quite a few are asexual. Even some animals and plants can reproduce without gametes. Cells of your body, for example, and bacteria serve as great examples of organisms that reproduce asexually. When a bacterial cell reproduces, it splits into two cells, each daughter cell a genetic replica of the mother cell. Such genetic replicas of cells are called clones. Though the methods of reproduction differ between sexual and asexual organisms, they each have something in common: DNA. Reproduction, whether it's sexual or asexual, is really nothing more than a method to transfer DNA to the next generation. Offspring of sexual organisms contain a mixture of both parents' DNA, whereas offspring of asexual organisms contain the DNA of the single parent. Let's consider a molecule of DNA, its structure, and how DNA can make copies of itself so it can be passed on to the next generation. From there, we'll look at a basic method of asexual reproduction called mitosis. Here's a molecule of DNA. DNA is famously called a double helix. Let's look at a slightly less confusing picture. Let's just look at the basic structure of DNA. Number one, we know it's made up DNA molecules. In fact, all nucleic acids are made up of repeating subunits called nucleotides. Nucleotides are the monomers of nucleic acids. And each nucleotide of a DNA molecule has three parts: a deoxyribose sugar, a phosphate group, and a nitrogenous base. As we see, there are three different nitrogenous bases. Four: A's, G's, C's, or T's. And of course, these stand for thymine, adenine, guanine, and cytosine. Let's look at a slightly different picture showing the same thing with the sugars. The S's stand for deoxyribose, of course, that's the name of the sugar. Bonded to each deoxyribose is a phosphate. And notice the repeating subunits of sugars and phosphates along one side of this molecule. Sugar phosphate, sugar phosphate, sugar phosphate, and so on. This is often referred to as the backbone of the molecule. We know that DNA is a double helix, so here's one side of the molecule, and here's the other side, the other helix. Likewise, it has a sugar phosphate backbone. Sugar phosphate, sugar phosphate, sugar phosphate. These are like the sides of a ladder, often compared to a ladder. If the sugar phosphate backbones are the sides of the ladder, then the rungs of the ladder are those nitrogenous bases: A's, G's, C's, and T's. Watson Crick and other scientists that working in the 1950s described this base pairing rule of DNA. For example, an adenine on one side of the molecule always pairs with a thymine on the other side. Likewise, a guanine will always pair with the cytosine. Now, I say always. We'll see in later chapters sometimes mutations happen when that base pairing rule gets messed up and it doesn't work the way it's supposed to. So we see base pairs. The pairs of bases are held together with hydrogen bonds between them. And these hydrogen bonds, as you might remember from a previous chapter, are quite easily broken. And that's a good thing, because DNA must make copies of itself for reproduction to be successful. For DNA replication to happen, those bonds, these hydrogen bonds, have to vanish. We'll watch a short video, and it shows DNA replication in more detail. But the things to be on the lookout for are some enzymes. One enzyme called helicase. That's a molecule that unzips the double helix. Another molecule that we see pictured here is called DNA polymerase. And once the DNA molecule has been unzipped, DNA polymerase is responsible for adding new nucleotides to the unzipped strand, resulting in, as we see here, 2 new molecules of DNA. Notice that each of these molecules of DNA contains a purple part of it, which originates from the original strand. And the artist has color-coded another strand red, indicating that's the strand that's recently been deposited by DNA polymerase. So let's look at that video.
To make perfect copies of our DNA, an enzyme first unwinds the 2 strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine. In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
As mentioned, life reproduces sexually or asexually. Though most eukaryotic organisms reproduce sexually, individual eukaryotic cells of an organism undergo asexual reproduction. The cell cycle describes the steps involved in such cellular reproduction. The cells of your skin, muscle, bone, organs, and other areas go through the cell cycle. The cell cycle is a topic of the upcoming micro lecture.